Thank you very much for the polite call. Dear colleagues, ladies and gentlemen, Professor Karikó Katalin, first of all, I'm really very glad that you are here. I think this is the question of the greatest importance today. You must have remembered that yesterday in Debrecen the weather was dark, hopeless, cloudy, some rain, some snow or a snow-like substance. And today, because of Katalin is here, the sun is shining, and I think that the scientific conclusion can easily be drawn that Debrecen in December is an ugly place, except when Katalin is here. So I think probably the second very important and interesting chapter is the talk you will deliver today. I thank you very much in advance. And as we clarified that we know each other for a very long time, I think the story is longer than 40 years. I think that's a prerequisite for the future and the promise for the future. Please deliver your talk.
Thank you. Ladies and gentlemen, members of the Faculty of Medicine or invited guests, dear students and our special guest, Professor Katalin Karikó. It is my great pleasure and privilege to welcome all of you to the presentation ceremony of the Debrecen Award for Molecular Medicine of the Faculty of Medicine. The Faculty of Medicine has a long tradition in encouraging basic and clinical research. Our eminent research groups strive to reach the front line of biomedical research and to push the boundaries of science by pursuing cutting-edge research. Our faculty also aims to strengthen its position as a regional center for international graduate and postgraduate training. As an acknowledgement of our leading role in the region, the European Union recognized us as a center of excellence in molecular medicine. We also feel that in this capacity, we are obliged to identify and recognize researchers who contributed to the field of molecular medicine. The international prize, the Debrecen Award for Molecular Medicine, is offered to recognize groundbreaking, extraordinary, and sustained contribution to biomedical research. The award acknowledges significant advances in the field of molecular medicine with demonstrated benefits applied to improved patient care. This year, the selection committee of the Debrecen Award for Molecular Medicine considered Professor Katalin Karikó as one of the nominees for the award. Now, I ask László Csernok, Vice Rector of the University of Debrecen, to announce the decision of the 2021 Debrecen Award for Molecular Medicine.
Ladies and gentlemen, dear guests, it is my duty and privilege to report that the professors of the Faculty of Medicine have decided by secret vote that in 2021 the Debrecen Award for Molecular Medicine should be presented to Professor Katalin Karikó. Professor Karikó is a biochemist and researcher, best known for her contributions to mRNA technology and the COVID-19 vaccines. Professor Karikó and her collaborator Drew Weissman invented the modified mRNA technology used in Pfizer BioNTech's and Moderna's vaccines to prevent COVID-19 infection. The novel mRNA technology made mRNA safe, effective, and practical for use as vaccine against COVID-19 and other human diseases. Now I ask Zoltán Pap, Vice Dean for Scientific Affairs, to highlight some relevant facts and data pertaining to the biography of Professor Kaliko Katalin.
Ladies and gentlemen, Katalin Karikó grew up in Kisújszállás and attended Móricz Zsigmond Református Gymnasium. She received her bachelor's degree in biology in 1978 and her doctorate in biochemistry in 1982 from the University of Szeged. Thereafter, she was working at the Biological Research Center of the Hungarian Academy of Sciences in Szeged before moving to the United States in 1985. Professor Katalin Karikó's research has focused for decades on RNA-mediated mechanisms with the ultimate goal of developing in vitro transcribed mRNA for protein therapy. She investigated RNA-mediated immune activation and co-discovered with pan-medicine colleague Drew Weissman the nucleoside modifications suppress the immunogenicity of RNA, which has widened the therapeutic potential of mRNA in treating diseases. Professor Karikó and Professor Weissman found a way to modify mRNA and later developed a delivery technique to package the mRNA in lipid nanoparticles. This made it possible for mRNA to reach the proper part of the body and trigger an immune response to fight disease. Since mRNA synthesized with this laboratory technology is extremely stable, further modifications resulted in an effective tool for achieving high output production of the coded protein in the targeted cells. This achievement opened wide potential opportunities to protein supplementation and the treatment of numerous genetic disorders, even in experimental oncological therapies. She owns or co-owns 14 patents, mostly through a project company, RNARX. She became also the CEO of. Professor Katalin Karikó is a senior vice president at the BioNTech and adjunct professor of neurosurgery at the University of Pennsylvania. Professor Karikó was recently honored with the Breakthrough Prize of Life Sciences, the Princess of Asturias Award, the Lasker Prize, Széchenyi Prize, the Wilshire Prize for Excellence in Biotechnology, and many, many others. She continues to work on new therapeutic applications of mRNA therapy. Google Scholar keeps track of Professor Karikó's 200+ peer-reviewed papers. There are more than 15,000 citations and an H-index of 57.
Dear Professor Katalin Karikó, now you are kindly requested to come forward to this table for the conferment of the Debrecen Award for Molecular Medicine. Ladies and gentlemen, Professor Katalin Karikó will now deliver her inaugural lecture.
Dear colleagues, ladies and gentlemen, students, I didn't try to mislead you that the title is different. I just thought that first I would say something about what kind of bridge I had towards University of Debrecen. As you heard, I grew up in Kisújszállás, which is just 80 kilometers from here, and thanks to my parents, I learned that hard work is part of life, and followed my sister's footsteps, older sister, and I just followed her in many different ways. But I didn't get here from the 80 kilometers, not from Kisújszállás. First, we all went actually to America. And this is where I met all of my friends who actually studied here at University of Debrecen at that time his daughter. And I came here first time to give a lecture in 1995 when Esther Csánki invited me and István Édes organized that meeting. Yeah, I was here much younger at that time, and gave a lecture about urokinase receptors. So that was more than 25 years ago, and came back in 2011 when Éva Remenyik and István Juhász invited me to be part of the Department of Dermatology program. And believe it or not, this was the first time in Hungary was used nucleoside-modified RNA. And we worked together and made a special RNA coding for cyclobutan pyrimidine dimer photolase and try to understand different kind of effect of UV makes on the skin. And one of the students was here, Gábor Boros, who worked on this project, and he helped later, came to BioNTech together with Gábor Tabás Szabó. Both of them get their education here and get their advanced degree here at the University of Debrecen. So I learned during the couple of years at BioNTech a lot about your university, and they helped to advance the messenger RNA program and actually helped the vaccine project, both of them very much. And thanks to them, my Hungarian is getting better also. So, but right now you cannot hear that. So they helped this program to advance. So, because what I am talking about today is how the mRNA was developed for therapy, and which we already know that not happened at the light speed. Seems like the overnight was a couple of decades of overnight. And I would like to tell you about this process where many scientists, those who are not with us anymore, advanced the science. They couldn't see that where it will end up. And the mRNA was discovered 60 years ago, and it took 60 years that we finally have messenger RNA as an approved drug in an LNP formulated mRNA vaccine forms. But during these years, a lot of things happened, a lot of scientists worked on it, and when 1978 first mRNA was isolated and delivered into mammalian cells. In 1984 was important when mRNA could be synthesized in vitro and we could generate RNA in our dish. And the people hope was up in 1990 when first time showed that mRNA injected to the animal could be translated. With my colleague, 2000, we discovered that actually the mRNA we are making in the tube synthetically actually is inflammatory, and took us quite a while when we figured out how to make it non-inflammatory, which finally get to formulated and get as an approved project. Even before the mRNA was discovered, actually people work with RNA, and was very important when they discovered that mRNA can carry all of the, or that is a viral RNA, can carry all of the information which was required to make a full virus. And these studies were done in tobacco virus, and they phenol extracted the RNA and brushed into the leaf of the tobacco leaf, and then they found that full functional infectious viruses were formed. So that was a very important point when recognizing that The messenger RNA carries all of this viral messenger RNA, all of the information needed to generate a virus. Of course, they ask what all of the protein is doing, and of course it was obvious that they are helping to enter to the cell in case of mammalian cells, and also protecting the RNA, which was very fragile. Of course, the scientist was trying to understand that how the proteins are produced when the genetic information was, all of them in the DNA, in the form of chromosome, but the protein was made in the cytoplasm and on the ribosome. And they tried to, and did a lot of experiments. They suspected that maybe this RNA will be the answer, this messenger RNA. And of course, they found in the ribosome an RNA, and they thought that that will be it. So they did an experiment in Spirin and colleagues in Russia, and they isolated the DNA from different bacteria, and they found that different nucleic acid composition were there, and when they isolated the RNA, they found that the nucleoside composition was the same. So they recognized that ribosomal RNA is not the messenger RNA. And I'm just telling you all of this because in every point in life, limited information is available where the scientists can work. And they do a limited number of experiments, but they are doing a lot of thinking, and it is just fascinating me always read a paper from older time. But by 1961, scientists discovered that there is a molecule, and this is the messenger RNA, which has very short half-life. 2 independent papers in Nature published as a very labile molecule. But this is the information flow, as we heard already, the DNA, the RNA, and the protein. Of course, identifying RNA is one thing, but making it is not. It's still waited for 20-something years. So what the scientists could do is to isolate. And what they did, they isolated from reticulocytes, which is the precursor of red blood cells, and they could isolate this globin mRNA. And when they isolated the globin RNA, first they translated it in a reticulocyte system, and then they injected, and other scientists, frog oocyte, and they found that this RNA can be translated to a protein which was encoded by. And an important milestone was 1978, when the same isolated RNA was wrapped into the lipid liposome and delivered in primary mammalian cells, and demonstrated that an isolated RNA delivered to mammalian cells can code for the protein which can be detected. So this was when actually 78 first mRNA transfection successfully accomplished, 2 independent laboratories. So this was the experiment actually my colleague Éva Kondorosi and Ernő Duda was reading about, and they were encouraged, and they started to use liposome for delivering nuclei and other materials to mammalian cells. And that time as an undergraduate, I joined the lipid lab, and Eva and Ernő were ready to isolate some phospholipid, and that's why they came to the lipid lab. And so we had limited resources, so Ernő had to go to the slaughterhouse to came and bring a co-brain back, because we had an old procedure how to make phospholipids. So before we could do the experiment, we had to work a whole week just to get the phospholipids. And so we wrapped the plasmid DNA into this, with this phospholipids and delivered to the mammalian cells, and the plasmid-coded protein was detected. And this is, for me, as started my career, it was just so impressive that such a novel things we can do, just deliver and the protein is made. And even today, when I look back, they were so ahead of their time, doing this kind of experiment. But for me, it was everything, of course. For a young scientist, everything is so novel and so exciting. And so for me, it determined very much what I have done maybe in the next 40 years. But from the lipid lab, I started to make my PhD in the RNA lab in the Biological Research Center, and Jenő Thomas, organic chemist, he made cap analogues for the identifying what is the 5' end of the mRNA. So he was doing it in 1975, helped those scientists, and I learned in the first day on job that messenger RNA has a cap structure on it. And I started to work there with János Ludwig, and we synthesized different molecules. And János was also a graduate student, and I learned a lot from him. And even today, we are still working together. Our role was in that time not to make a messenger RNA, but a small antiviral molecule, which was linked by 2', 5'. And this molecule, which was induced by, was generated by an enzyme which was induced by the interferon, and in response to double-stranded RNA, this small molecule was made, which could bind to the enzyme and degrade what was at that time the hypothesis, the viral RNA. I have to mention that not just, mention to the young scientists to follow what the colleagues are doing on the different field, but also what you started to work with in early on. And for example, the OAS, which is this enzyme, can make this molecule. Even today, we know that how important for developing a response to the COVID infection. But we kind of work in these different small molecules. But meanwhile, scientists in the other corner of the world, in Harvard University, they discovered that how they can synthesize RNA from using a phage polymerase. And so what they did, human beta interferon coding sequence, they put in this plasmid construct and put all of the enzymes and the nucleotides and the capping enzyme, and generated an RNA, which very much looked like our RNA in our body. And what they did, they injected to frog oocyte, and which was cultured in the medium and collecting the medium, and they demonstrated that those were functional and inhibited viral infection. So this was the first time when, in 1984, could be made an RNA for what these scientists wanted to, and demonstrate that it translates to a functional protein. So this was a major milestone in the mRNA therapy development. At the end of this 1980s, a lot of things happened and a lot of progress was made. And what was important is that scientists introduced this lipofectin, which helped complex the RNA, which was RNA is negatively charged, the lipofectin is positively charged, and made it very easy for anybody, even for me, to make mRNA and deliver to the cell, and a couple of hours later analyze the protein which the RNA coded for. So a lot of scientists started to use this technology for that. And some study already, end of the here in 89, already delivered RNA for mammalian cells. In 1990, when in vivo delivery was first reported, because this lipofectin, it helped a lot of scientists to make experiments on the cultured cells, but those were very toxic in animals. So most of the animal study, actually, they used naked RNA, which is just generated the RNA and injected to the animal. And in 1992, already they could treat a model animal delivering vasopressin RNA, and the animal were not diabetic anymore. But subsequently, my interest was always to use mRNA for some coding for some therapeutic protein, but most of the scientists actually were using for vaccine the RNA in the early 90s. 2 papers were published where scientists used for generating vaccine for influenza. Scientists in France, they used actually liposome, similar what we have done in a biological research center, and the others is just self-amplifying RNA and naked RNA injected to the animals. But it's about that. That was it for infectious disease vaccine development. From that on, in the mid-90s and later on, all of the messenger RNA vaccine project was focusing cancer treatment. And first in 95 was the first paper was published, and 96 Eli Gilboa from Duke University, he used human dendritic cells, delivered messenger RNA, which was isolated from the tumor, and they amplified by RT-PCR, and they actually, in 1997, they established the first messenger RNA company, Merix. And later it became Argos when Ralph Steinman, who received the same award from University Debrecen, Ralph Steinman joined, and they made Argos, and this company was using human dendritic cells, which they generated from the patient, and added messenger RNA, tumor-specific RNA coding, tumor-specific protein coding RNA, and they did clinical studies. So the longest history of the messenger RNA application is cancer vaccine related. While these scientists were working on cancer-related studies, I joined cardiology at the University of Pennsylvania when Elliot Barnathan hired me for a faculty position, and he expected me to establish myself, which never happened, but he was very enthusiastic and supported all my effort to make messenger RNA for therapy. And in cardiology, we were interested in intimal thickening, and so we cloned the urokinase receptor and made a messenger RNA coding for this receptor. And this receptor is so heavily modified, glycosylated, GPI anchored to the membrane. And what we surprised is, so all of this is needed for functionality, and what we surprised is that we delivered the RNA, and this highly post-translationally modified receptor was made, was functional, and it kind of felt that, okay, the messenger RNA will be good for something. And of course, we could do it only on cells, so we thought that maybe for cell therapy, we can use the RNA, and started to thinking about like using the RNA for bone marrow transplantation, where maybe extending the tip of the chromosome with telomerase mRNA, we could expand and help the cell to proliferate more heavily. And we were doing these experiments, and we kind of lost the position, but I was rescued by David Langer, a neurosurgeon resident, when he convinced the chairman that neurosurgery needs a molecular biologist and they should give a lab space. And so this was actually the laboratory I was doing for 18 years, all of the mRNA there, did the tissue culturing, and most of the time I was in a one-man show, so I worked everything myself, and we tried to do and come up with therapies for treating stroke patients. Since David was a physician, and he explained always that what he can do, what he can reach tissues or vessels, and I said what I can do, and we tried to work together, and we did a lot of work with inducible nitric oxide synthase, because we thought that dilating the blood vessel after subarachnoid hemorrhage, when it is constructing the blood vessel and stop the blood flow, would be beneficial for the stroke patient. We also did many of these antioxidant enzyme coding RNA work, because at that point was considered that those are responsible for oxidative damage, is responsible for many of the damage the stroke patient has. And later in my final effort, when I tried to get grant, my idea was that whether the stroke patient suffering a lot of cell damage, and the DNA, the RNA, everything which is coming released from the dying cells, and actually this is instigating the inflammation and other process, and what we should do is catabolizing those damaging debris molecules. And so we tried things, and we traveled around the country who had different model animals, but still, the major problem was always in the mRNA therapy, that the amount of protein was too small to make biological effect, and the RNA was too labile. And for the investors who we tried to get money, for them the problem was that the shelf life, there were no shelf life, that you could take out aliquots and characterize and go back a year later and another aliquot to take. So there were a lot of area that we had to improve. But one thing, what we did not know, that the RNA we are making actually is inflammatory. For that one, I had to meet Drew Weissman, who was an immunologist, and he wanted to develop a vaccine. And famously, we met in the copy machine, and I kind of like to brag about things, and I said, you know, I can make good RNA, and so he took on my suggestion and then tested out the RNA I made for him, and then he was interested in HIV vaccine, so I made a GAG, HIV-specific mRNA, and he was very happy that this RNA is so immunogenic, activated all of the markers, and also so much protein was made, and of course I was so sad when learned that the RNA is so inflammatory and immunogenic. So together we started to think that why this RNA we are making is so problematic and immunogenic. Could it be that we are delivering from outside, and then maybe the body, the immune cells, recognizing as foreign invaders? So we did an experiment where different compartment of mammalian RNA, bacterial RNA, we isolated, and we put in this human dendritic cells and measured that whether inflammation was induced. And this is this purple is when I made the in vitro transcribed RNA. That was the most inflammatory, most TNF-alpha was induced, but some of the RNA we isolated were less inflammatory, but surprisingly this tRNA did not induce any inflammation. And knowing that the tRNA has a lot of nucleoside modification, we started to suspect that maybe this could be the reason why it is not immunogenic. So we were thinking that we might have to make an mRNA which has this kind of modification. But the dilemma was there, that how we could do it. We could make an RNA from the 4 basic nucleotides, as in our own body is doing, and with the enzymes post-transcriptionally incorporate this kind of modification. But I don't want to scare you really, but there are a lot of modification there, just shown here. And the enzymes which can incorporate were not known. So we didn't know that some of them, not that you cannot order, you know, we didn't even know that which enzyme is doing. So that kind of way to produce as our body is producing the modified RNA was not feasible. So we had to find an alternative way. And what was that we purchased nucleotide triphosphate, which already had modification on it. We purchased 10 that was commercially available, and we tried to hope for that the RNA polymerase will incorporate. And in case of 5 of these nucleotide triphosphate, we could generate messenger RNA. And now that the RNA was made, we want to know that is it really less immunogenic, and test it similarly as the isolated RNA we did and measure the inflammatory response. And we found that some of them were almost as inflammatory as the conventional RNA, but some of them did not induce any TNF-alpha. When we looked at closer, we could see that all of those who did not induce any inflammation actually had modified uridine in that. So then we asked the question, nature selected uridine to detect as a foreignness, and seemed like that, but we did not know at this point. But the next question was, if we want to make a non-immunogenic RNA for therapy, we need also this RNA to be translatable. So that was the next step when we demonstrated that at least the 2 of these could be translated. One of the RNA containing 2-thiouridine was not translatable, but surprisingly, the pseudouridine containing RNA translated 10 times more protein. Again, something we could not understand why nature created like that. But definitely we were very happy that finally we have an RNA which is non-immunogenic, which so much protein is made, and finally we could proceed. 10 years later, scientists discovered actually that the uridine, in the case of toll-like receptor 8, which is a sensor of the immune sensors in the endosome, present in the immune cells, even the uridine, just the nucleoside itself can help dimerize and get activated the toll-8, and identify that how the uridine is performing this process with the, on the crystal structure. So there was an explanation here for that. So we want to know that whether this pseudouridine containing RNA, whether it is translating to a functional protein. So with my colleague Hiromi Muramatsu, who was willing to spend the Sunday and Saturday with hundreds of mice, we try to evaluate injected and taking tiny aliquot of blood so that the animals could be followed for weeks. And we identified that pseudouridine-containing RNA translates, and here we used erythropoietin-coding mRNA, and we could detect that translate very well, and the protein was detectable for 4 days. Experiment we also showed that after injecting the mRNA for erythropoietin, 30 minutes later we already could measure in the blood circulation. So the RNA translates instantly. And whereas when we had here just the protein itself, this has half 2 hours half-life is degrade, and the uridine-containing RNA was also detectable short time. Of course, the most important thing was that this modification resulted that no interferon, no inflammation was produced by this nucleoside-modified RNA. And when we injected this RNA, the EPO-RNA to the animals, we could see the hematocrit increase, so the biological effect was there. And when we injected weekly, once a very small amount, who is in the laboratory would know that what is it, 0.1 microgram of RNA, how small amount is that. But it was sufficient, translated to the protein, and because it was continuously translated and was present, so made sure that the red blood cells are formed. So we were happy that finally we found a disease form, which is for anemia, let's say, and where we can deliver an mRNA which can have therapeutic effect. But we had to leave, and with Hiromi, with my colleague, we decided that we will go to Germany, and we will introduce the nucleoside-modified mRNA somehow to the clinic. So we were on a mission, and there were no weekend, nothing, and we just performing experiment after experiment to figure out what should be. First of all, 2 years we spent just to figure out that what we have done at the university at the bench, how we can make it for a scalable, industrial, scalable manner. And so every procedures, let's say precipitation had to be eliminated because it is not feasible to do for large scale. And thanks to Ugur Zahin, he's the CEO of BioNTech, he gave us this opportunity to work there and introduce the nucleoside-modified RNA to the clinic. And we did a lot of experiment. Here I'm just showing you one that bispecific antibody encoding mRNA we are using. These were animal studies. The importance is that what is the advantage of mRNA versus the recombinant protein, which might be already in the clinic, is that the patient will make the protein from the mRNA, which means that it will be much cheaper because the purification and other procedure didn't have to be taken place. So many antibody, which is expensive, can be used, can be applied for and would be more affordable for people. The other thing is, for example, here is the bispecific antibody. Is half-life is just 2 hours. So the patient had to have a pump and with continuously pumping into their system the protein. But when we deliver the RNA, we just delivered here, you know, weekly, once a week or less, because the protein is continuously made for a couple of days. So that is the major advantage in this case. And of course, even if the product would be finally a protein product, but screening, performing screening and others would be much cheaper to do with RNA, because ordering a template is, and this is very affordable and cheap, and generating an RNA in 2 hours and delivering to the cell, and half an hour later you can analyze the protein. So it is very quick, and for screening purposes. So what we have done in BioNTech and other scientists as well is they start to optimize the RNA to make sure that more protein is made from it. So when we have a wild-type RNA, we have a small amount of protein, maybe a shorter period of time. But when you optimize the codon or the different element of the RNA, which is a non-translated region, then you get longer or higher protein production and longer. And when you combine all of these optimization and you purify it and modify it, then really you can extend the translation level and extend the time that protein is produced. And just mentioned about the codon optimization that in our case, if we are saying that the goal of the codon optimization is to eliminate this inflammatory uridine, for example, then of course we can just replace the uridine with pseudouridine and we can have a product which is not immunogenic. But somebody might can do it by selecting different codons, like, for example, the CureVac project. They selected GC-rich sequences and eliminated as many uridine as possible so that the RNA will be less immunogenic. With this optimization is one problem is that we have half of the amino acid cannot be coded without uridine. So in every time, and one of the amino acid is leucine, which is quite frequent, so every time your RNA will be depending, the immunogenicity depending on what will be the amino acid composition. So if more amino acid there, which requires uridine to code, then the RNA will be more immunogenic. My colleague, Norbert Pardy, who joined us in 2011, he focused and developing vaccine together with Drew Weissman, and at the beginning Norbert worked with me, and he did a very important contribution to the vaccine development, because he was the who started to use the lipid nanoparticle to wrap the messenger RNA, which had a nucleoside modification. At that time, we did not know that this lipid nanoparticle, why it is so critical for the vaccine. Today, or actually last week already, it was published that the lipid nanoparticle, one component is the adjuvant. And that is why it is so effective, this vaccine. The messenger RNA is not activating anything, but one component, the ionizable lipid, does. In the first experiment, actually, which Norbert published in Nature, was when he did a Zika virus-specific messenger RNA, and this messenger RNA was the first that a lipid nanoparticle was wrapped, and he vaccinated animals, including monkeys, with it. And what was in 2017 the major surprise that whatever amount was needed to protect a monkey from infection, from Zika, this was sufficient also, like the same amount in a mouse, and didn't need it to scale up. Other people previously tried to use gene-based vaccines, like the DNA-based, but they had to unproportionally inject more and more of the genetic material. But here, with the mRNA, it seemed that a very small amount was very efficient, and that is what shown here, that even 50 microgram was sufficient to fully protect these monkeys. So subsequently, Norbert did also herpes simplex, HIV, influenza, and SARS-CoV-2 vaccines in a laboratory animal, and tested and did major discoveries about the mechanism how this high level of antibody is generated. So where we are today, of course everybody thinks that now that the vaccine was developed and now that there is a future coming for the RNA. Actually the future was already in the past, because messenger RNA was advanced clinical trials for many different applications. The first one initiated from Harvard University and AstraZeneca developed for heart failure treatment, which the human phase 2 trial just ended recently. And in that case, during bypass surgeries, the muscle of the heart were injected VEGF-A mRNA, which generated new blood vessels. And the study resulted that showed that those who were get this RNA injection, the capacity of the heart was significantly improved. So that was actually the most advanced, and we expected those scientists who are gathered mRNA therapy meeting from 2013 that this will be the first one which will be approved. BioNTech also has an advanced phase 2 trial for individualized cancer vaccine, and other application like generating RNA, which codes for cytokines, which injected to the tumors to generate, to convert the cold tumor to hot tumor. Actually, one of the projects I participated, initiated at BioNTech. And in this case, the mechanism is not, the RNA is not a vaccine, but the RNA is the cytokines, which is coding for the cytokines, injected to the tumor, which is close to the surface, like melanoma or head and neck cancer, and then inviting the immune cells to this vicinity of the tumor and processing it, and then due to the circulation, finding a metastatic spot where the similar cells are there and destroy that. So this is human trial is ongoing with Sanofi. And it seems that, you know, always criticized mRNA is so labeled, they're great, but it seems now today that maybe the promise of gene therapy will be also solved by the mRNA. And for this one, we already know Intellia used Cas9, this genome editing enzyme, and it delivered to the patient as an mRNA, nucleoside-modified RNA to the liver, and transthyretin amyloidosis suffering patient, 3 of them were permanently cured. So more is coming on our field. Preclinical studies are going, vaccination, actually these vaccination also going on human. We know that the influenza trial just phase 1 trial ended, but scientists already, if you would attend to the RNA meeting, 3 years ago, you already could hear that scientists developed mRNA for malaria vaccination and many other areas. Here in Semmelweis University also they are using now with Norbert Pardy help together this VEGF-C mRNA, for example, that generate new lymphoid vessels and helping to alleviate edema. So many applications, as maybe you are scientists and you have some kind of problem that maybe acute problem that could be delivering mRNA for treatment if you know what should be overexpressed, because this is overexpressing a protein is related. And of course, you know, the most important breakthrough was and this year and last year is that the vaccine was generated from messenger RNA, and we learned that it was a short period of time from the knowledge of the viral sequences when it made public the vaccine was produced. If this information, let's say, would be like earlier, like even just, let's say, 20 years ago or 10 years ago, it would not happen this way, because gene synthesis, for example, was not solved up until the early 2000s, so that the information would come at that point. Everybody who wants to develop a vaccine would need a package coming from or containing some viral genetic material, because you just couldn't order a gene, just like you do today. And so I don't go to details, because many of us, you know, watch the television. Each of the step when was reported that different phase trial ended, and eventually in this year, the FDA approved the vaccine for Pfizer and BioNTech is fully approved. So with that, so I try to tell you that so many scientists, everybody I mentioned who was part of this process, who contributed to the science, and so that's when I would like to thank you for your attention, just to make sure that you know that so many people, I was happy that I was one of them, but many, many scientists, my present colleagues, people who are working in laboratories, working in the companies, running the clinical trial, the heroes of physicians who were helping the patient, the people who are volunteered for the trial, all of us, all of these people contributed, and so I would like to thank all of them, and to you also, because you worked very hard and still today to have the patient. Thank you.
Thank you very much. I declare the ceremony closed. Let me just thank you again, and may I wish you continued success in your private life and in your research. Thank you very much again for coming. I declare the ceremony closed. Thank you for your participation.
Thank you.